Scientists are building cells from scratch. Not growing them, building them, piece by piece, in a lab. A synthetic cell is an artificial structure built to copy what a living cell does: grow, react, even divide. This isn't just a lab trick. It touches the biggest question in biology: what actually separates living from non-living? The idea isn't new. Scientists dreamed of building life since the 1990s, but the tools only caught up recently. There are two ways scientists build them: top-down and bottom-up. Top-down means taking a real living cell and stripping away everything except the genes it truly needs to survive. The J. Craig Venter Institute built Syn3 cell with just 473 genes, the smallest genome known to sustain life. By seeing what's left when you remove everything non-essential, scientists learn what life's true requirements really are. Bottom-up is the opposite, building a cell from non-living parts: lipids, proteins, and DNA, assembled from nothing. First, a boundary. Lipid molecules naturally curl into tiny bubbles called vesicles, a cell's very first wall. Lipids do this on their own. Their water-loving heads and water-fearing tails naturally push them into a sphere. Scientists often use giant unilamellar vesicles, bubbles large enough to see under a regular microscope.
Inside, scientists add machinery for energy production, letting the synthetic cell power its own internal reactions.
They also add a pure system, purified molecular machinery that reads DNA and builds proteins from scratch. This means the synthetic cell's DNA works like software. Scientists can rewrite it to change what the cell does. Some synthetic cells can even grow larger and split into two, the earliest sign of lifelike behavior. But division is tricky. Without a natural cell's precise machinery, synthetic cells often split unevenly or not at all. Some synthetic cells can even sense their surroundings, reacting to light, chemicals, or changes in temperature. One reason to build them? Understanding how life began on Earth billions of years ago, from simple molecules. Drug delivery. Synthetic cells can carry medicine directly to a specific spot in the body. Reason 3, biomanufacturing. Simplified synthetic cells can produce his chemicals or proteins more efficiently than natural ones. Reason 4, disease modeling. Synthetic cells can mimic sick cells, letting scientists safely test new treatments. Reason 5, biosensors. Synthetic cells can detect pollutants or toxins, acting as living environmental alarms. To build these precisely, scientists use microfluidics, chips with channels thinner than a human hair. These chips generate thousands of uniform droplets per second, each one a potential synthetic cell in the making. This isn't one lab's project. Groups like Build a Cell and the European Synthetic Cell Initiative collaborate worldwide. But no synthetic cell today is fully alive. Most can only do one or two of these things at once. This raises real questions. How do we safely contain synthetic life, and who decides how it's used? Researchers are getting closer every year. Smarter membranes, better division, and cells that respond more like real ones. Science is inching closer to building life from scratch. Follow for more mind-bending science, explained simply.